name's Liane. I'm sure I know everyone here. And my talk today will be a little bit different than the last talk. So my project is a little more basic science heavy. So if there's any questions about that, or if I seem to skip over something that should have had more introduction or description, please let me know. So the title of my talk is Defining the Transcriptional Regulatory Network of Anti-Angiogenic Therapy Resistance in Glioblastoma. It's a mouthful. My mentor is Dr. Vineet Shaghi in the Department of Neurological Surgery. So an overview of my talk today, I'll do some background on glioblastoma, treatment, standard of care that is used for this disease, the introduction of anti-angiogenic therapy and why it's used in glioblastoma, and then talk a little bit about the development of resistance to this treatment, how we study that in the lab, and then I'll go into my project aims, my experimental design, and that should be it for today. So glioblastoma, I'll call it GBM most likely, is the most common primary brain malignancy in adults. However, the incidence rate is 3.19 cases per 100,000, so it's very rare. It is characterized by the World Health Organization grading classification. So if you remember, there are 4 classes. Class 1 is pretty benign. CNS malignancies, with grade 4 being the most aggressive. The grades are dependent on certain histological factors from biopsies, as well as clinically how aggressive and invasive the tumors are. Glioblastoma is grade 4, it's highly vascular, has a lot of neovascularization, nuclear atypia, high mitotic index. And despite all this extra vascularity, there is a high level of necrosis, usually centrally located in these tumors. Radiologically, they appear as ring-enhancing lesions in the parenchymal region of the brain. There's usually surrounding edema, and with contrast, they will heterogeneously enhance. And the prognosis is very poor, and recurrence is quite common. So to go a little bit more into the prognosis, the 2-year relative survival of all gliomas, which is shown here, basically this is data collected between 1973 and 2002, and you can see here with glioblastoma, this is just categorized by age at which they were diagnosed. You can see that the 2-year relative survival rates are quite low, ranging from 1.4% to about 28%. The median time to recurrence for these tumors after treatment is about 7 months. And the average length of survival after treatment is only 15 months from the time of diagnosis. Treatment options for glioblastoma. So the standard of care right now is based upon a trial that was done in Europe and was a randomized trial with an organization there as well as in Canada. And it includes maximal surgical resection, obviously with the goal to preserve as much neurologic function as possible, plus daily chemotherapy and radiation. The chemotherapy is usually temozolomide, and then maintenance with chemotherapy thereafter once the radiation therapy is done. Recurrence is common, as I mentioned, despite optimal management. And once recurrence does happen, it's associated with even worse outcomes. So 6-month progression-free survival rate of 9% and 12-month overall survival of rate of only 14%. This Kaplan-Meier curve here, the gray line, it says yes here, is basically patients who received the standard of care therapy, so surgical resection plus chemo and radiation. No is patients who did not receive that. And you can see that even despite optimal management, prognosis is not good. And then in this picture here, there's a GBM here. This was pre-surgery. In B, you can see the cavity after surgery. And then C here is 6 months after the surgery. You can see that it's the tumor has recurred in the tumor site, but also in an additional site. They went back in, resected both of those, and then 3 months later, you can see still remaining tumor. So, given this, there's been an obvious need for better treatment options, additional options in managing glioblastoma. And given that they're highly vascularized and they have increased expression of pro-angiogenic factors such as VEGF, it was hypothesized that using anti-angiogenic therapies and targeting that pathway could provide a significant benefit for these patients. One such therapy is bevacizumab. I will call it Bev because it's a mouthful to say all the time. Bevacizumab is a humanized monoclonal antibody to the soluble VEGF-A, which is here. This little purple thing is supposed to be Bev. It's also called Avastin. If you've heard of Avastin, that's the drug name. So VEGF-A usually binds to its receptor, which is VEGF receptor 2, and then leads to increased angiogenesis. And the idea behind this antibody is that it does not allow for the binding of the ligand to its receptor and leading to downregulation of angiogenesis. So initial studies using Bev started in recurrent GBM, and they were quite encouraging and promising. They showed decreased tumor size, fewer blood vessels, and decreased neovascularization as they expected, and even increased progression-free survival rates. Multiple studies did show that was there, and because of that, let's see, I actually have some numbers here. So for the increased progression-free survival rates, when patients were given bevacizumab alone, the 6-month progression-free survival ranged between 29 to 42%. So if you remember back about 2 minutes, that's compared to 9% of patients without bevacizumab. So seeing that great of a difference in progression-free survival is pretty significant. You usually don't see that. And then when bevacizumab is used in combination with other chemotherapeutic agents or radiation, the progression-free survival is also much higher. So given that, in 2009, bevacizumab was actually, the approval of it was accelerated through the FDA from going from phase 2 trials to immediately available for use in recurrent glioblastoma. And then once that happened, its use and study for newly diagnosed glioblastoma kind of took off as well. In terms of newly diagnosed glioblastoma, there's were 2 studies that showed improvement in progression-free survival from 7.6 to 13.6 months, but they weren't seeing a really significant change in overall survival, 19.6 months versus 21.1 months. And I believe this study is referring to this Kaplan-Meier curve here, And this was in combination with standard of care treatment, so bevacizumab on top of chemotherapy and radiation. So there was initial excitement about this since there was some positive results. However, there's been fading enthusiasm about the use of bevacizumab for patients with glioblastoma because there's been variable results, and probably most recently in the New England Journal of Medicine, published in February, there was a big trial that was in there that showed that bevacizumab plus chemotherapy and radiation with maintenance chemo did not actually improve in terms of overall survival. And there was a slight improvement in progression-free survival from 7.6 to 10.7 months, but this actually wasn't significant for their criteria that they had set. They also found that there were significant side effects and toxicities associated with bevacizumab. One of the well-known side effects is hypertension. That's actually how they sometimes titrate or know if bevacizumab is actually at a appropriate level is the hypertension that the patients may have. But there's also bleeding, thromboembolic events as well. So this was a little frustrating because there was all this excitement about it, and now it seems like there's definite improvement in progression-free survival, but that's not really translating to overall survival in the patients. So what's going on here? There have been studies that have shown that a small portion of patients actually don't respond at all to anti-angiogenic therapy, and they're thought to have intrinsic resistance. But more strikingly is that despite initial response, approximately half of patients treated with bevacizumab will have tumor progression. And that patients who do have resistant tumors, so they responded but then reoccurred, they have a poor prognosis and do not respond well to additional therapies thereafter. So in this study, they found that if patients received bevacizumab before their initial surgical resection, so the solid black line, they actually had decreased overall survival from 40% to 6%, and 6-month progression-free survival of 0% to 41%. And as well down here, you can see that it's not showing up very well here for me. So this patient had a tumor here that was removed pre-bevacizumab, but once they were treated with bevacizumab, you can see that there is much more extensive progression of the disease. And that's something that's been seen frequently, is actually these tumors that are recurring after bevacizumab treatment are more invasive and aggressive. There are multiple proposed mechanisms for this resistance, involving transcriptional and post-translational modifications that allow evasion of this angiogenic blockade while the drug is still binding its target. So that's kind of unlike other therapy agents where the cells have found a way to either not allow the molecule to bind, but here VEGF is still being bound by bevacizumab and not acting on its target. So these mechanisms include changing in blood flow alterations, differences in hypoxia leading to different transcriptional regulation and different transcription factors, epithelial to mesenchymal transition, allowing for that more invasive tumor type, activation of cancer stem cells, recruitment of bone marrow-derived cells as well, and then changes in the dominant VEGF isoform. So how do we study this in the lab? The main way that most have been looking at it is taking the patient-derived tumor samples that are collected from the OR, and then using those in either mouse models or in vitro to study the resistance. The limitations with this, however, is that the supply is usually pretty low. There's only so much tumor that's available, and there are multiple labs that obviously want to utilize that material. And the other significant thing is that the development of resistance to therapy has already occurred. So understanding maybe what's driving those mechanisms or where the development of resistance is really starting can't be elucidated. So someone in the lab that I'm working in developed this alternative model, a xenograft model in mice of acquired resistance that allows for a more chronological and temporal analysis of resistance development. So just to give you a sense of how this works, so we took glioblastoma primary tumor cells, injected them into mice subcutaneously. It's a standard way of doing in vivo studies for this tumor model. And then the mice were either treated with a control antibody, just IgG, or treated with bevacizumab. And then these mice were considered generation 2. The first generation was prior to treatment with either therapy. Then the fastest growing tumor from each generation was repropagated to another set of mice. And the treatment was continued. So with the idea that the fastest growing tumor, particularly in this bevacizumab treated group, is going to be the one that's becoming resistant to the therapy. And this was actually continued on, I believe, around 11 generations at this point. So having this mechanism has actually allowed us to collect multiple samples to look at to identify where resistance may have more significantly developed and get a better sense of what's going on there. So my year-long project, my aim and main goal is to further characterize the transcriptional regulatory network that occurs in anti-angiogenic therapy resistance in glioblastoma. So other people in the lab are looking at more protein interactions using that model, and I'm going to be focusing on the transcription factors and the epigenetic landscape that is important for driving and maintaining this resistance development in this model. And then creating a regulatory cascade of transcription factors and target genes that are involved with the potential, hopefully, to identify new therapeutic targets that could be given in addition to bevacizumab to increase its efficacy and allow it to actually utilize to its fullest therapeutic potential, but also potentially find novel biomarkers that may be associated with this resistance development, so we can find it early. So my plan in order to do this, starting with identifying the epigenetic and transcriptional landscape in bevacizumab resistance, I will be using a technique called chromatin immunoprecipitation, or CHIP, that will probe for active sites of chromatin, so open areas of chromatin, where transcription is happening, and this is usually marked by acetylation at a certain histone, histone 3 at a lysine 27. After enriching for these areas, I will sequence that DNA to determine the differential gene activation between bevacizumab responsive and bevacizumab resistant tumors. And then I can use computational analysis and microarray expression data to identify transcription factors that may be candidate regulators of what's happening here. So I'm a very visual person, so I also provided a little workflow of how CHIP works. It's called CHIP-seq if you do chromatin precipitation and then sequence. This is your DNA with the chromatin bound, and this little circle is a transcription factor, and these are different modifications on the histone. So you first cross-link your sample to bind any DNA that is there to your, dividing protein that is there to your DNA. You then shear the chromatin randomly, either with sonication or different enzymes to break it up. And then you immunoprecipitate with an antibody either directed against your histone modification or a specific transcription factor. That antibody will bind. You can then use either magnetic beads or other forms of beads to then just hold on to those portions of the DNA that are bound to the antibody, and then you can purify your DNA fragments. You reverse the cross-linking, construct a library of those DNA fragments, and then sequence the DNA. So once it's been sequenced, you can align these enriched sequences to the whole genome. You align them and then you map them to the whole genome, and it creates these peaks, which in the bioinformatics world is called peak calling. And this peak here is equivalent to what you're seeing here. This is a whole genome map or a portion of a whole genome map, and you can see where the enriched sequence or the enriched fragments of DNA are, and then you can then determine what genes are being turned on. And then using other computational analysis, you can determine if there are any overrepresented motifs for particular transcription factors, because transcription factors bind to DNA in a very particular sequence, and then also find transcriptional start sites to further determine what transcription factors may be involved. So once that has been done and a list of transcription factors have been identified as potential candidates via ChIP-seq, I will compare that to expression data from a microarray that we already have that is currently being analyzed now to identify potential candidate regulators of this resistance. And then using in vitro and in vivo functional assays, so looking at cell survival, proliferation, migration, and invasion, I will determine the transcription factor, but more likely set of transcription factors that can convert this resistance in cells. Once that has been done, I can perform ChIP again, but now using antibodies that are specifically targeting those transcription factors to figure out what their regulatory targets are, and figure out which genes are being activated, and then can create a regulatory map of what is going on in the resistance. So if you remember back to my little mouse picture, I have multiple generations that I can look at. I'm starting with one of the later generations in a place where I know that resistance has been established and developed, and will compare that to its counterpart in the same generation. But then what I'm hoping to do, if there's time, is actually go to an earlier generation where qualitatively people in the lab said that they noticed that there started to be a difference between generations on how quickly the tumors were growing, and see if I can maybe identify what the drivers of the resistance are, not just the ones that are maintaining resistance in these cell lines. So with that is my plan. Right now I'm in the process of optimizing my ChIP protocol, as it is a slightly finicky protocol. And I hope at my next whip I can show you some actual data from that. Any questions about it?
So I'm just curious, is the idea to ultimately give some sort of adjunctive therapy to bevacizumab, or to identify people that would and would not benefit from treatment with it in the first place?
That I think depends a little bit on what we find. I think ultimately we would like to be able to do both. to see if there are certain biomarkers that can help us determine who would benefit most from receiving the treatment, particularly because it is very expensive, as well as it does have these high toxicities. So if we could just utilize it in patients who would be most useful for, that would be best. If we could also find potential targets upstream of the upstream of the resistance development that might cut off that from happening, we would also consider utilizing it as well.
One thing I was wondering about was that, so the earlier studies that you had shown, bevacizumab was used in conjunction with the existing multiple sets of chemotherapy. And I know that in science you have to kind of focus on one question, but I guess how do you imagine that this will inform the real life situation where you're throwing 4 different medications?
Yeah, so it definitely would make it a much more complicated picture. And as I was mentioning before, there's actually, there are many proposed mechanisms to resistance that probably involve processes with other treatments, and it's probably a combination of all of those mechanisms that's happening, not just one. We do have expression array data from a series of patients who were resistant to bevacizumab. We had samples from prior to treatment and then after, and so we would like to compare what we find to that expression data as well to see if there is a difference. Most likely what I'm expecting is that our model will probably be more simplified and will really only give us a sense of one of those proposed mechanisms.
The other question was whether it was mouse GBM or human GBM that's xenograft.
It's human GBM.
So, just to be clear that I understand, it sounds like you give them the bevacizumab, a lot of people did better, then somehow, I didn't really see the pictures in detail, but whatever the mechanism is, the tumor kind of evolves and becomes resistant. So then you're looking at sort of end stage, these resistant ones, what their gene transcription and characteristics are like, but theoretically that's already very end stage, right? And if you want to looking at transcription factors as a unique source of information, I feel like you'd have to have a baseline comparison to before there's resistance developed. So I was curious if there's any other people looking at transcription factors within, initial diagnosis, and are there any therapies that target, can you target transcription factors, those gene sites specifically, or is transcription factors more to identify, like the CHIP stuff, is it to identify the genes that are implicated, or is it actually a therapeutic target?
Right now it's to identify the genes that are implicated, but with the potential that they may be therapeutic targets. And to kind of go back to your other question, I didn't explain this very well. So in order for CHIP to work, I need to have something to compare it to. So I will be comparing the resistant model versus the model that received control, the control antibody. So in addition to seeing what the activated genes are in the resistance model, I can also turn around and look at it and see maybe there's some suppression going on, or maybe that's causing this resistance as well.
Okay. And are you guys banking all of the original size tumor in like a bank or something that you go back and sequence afterwards once you've identified your target
We
genes,
do, we don't have we have our magic, but like that's, there are tumor samples available for us to use. However, that's the idea between, or behind the xenograft model was that the sample would be much more abundant than patient tissue, but it's actually limited as well, because you can't really have very large tumors on these little mice.
Kind of along a similar vein, I'm wondering if your control would be better, or if you wouldn't be better served using a control from the earlier generation of your GBM mouse that is treated with bevacizumab, because one of the problems with multiple generations of tumors is that they can grow, into whatever.